Welcome to another episode of The Scientist's Point of View, where I share important facts for you to understand our test properly. So you see this test at the top of the screen. Yes, this test, Widal. It is the enemy of diagnosing typhoid accurately. The WHO has even said that they do not find it reliable since the 1980s. That's even before the Gen Zs were born. But for some reason, Nigerians are still conducting this test. Please, let's stop. The recommended tests by the WHO are two tests, which are stool MCS and blood culture. Salmonella is what causes typhoid fever and is transmitted through contaminated feces in food and water. But this doesn't mean that if you've had typhoid, someone has pooped in your food or water. What this means is whoever made your food didn't disinfect their hands properly after doing the number 2. As we can already see, the mode of transmission for typhoid is through the feces. So it only makes sense why the WHO recommend stool MCS as one of the most effective ways to diagnose typhoid fever. So the MCS in stool MCS means microscopy, culture, and sensitivity, which will be explained briefly in this video. But before that even, the sample needs to be collected correctly. When a doctor sends you for this test, you're typically given a stool container like this. So there's a spoon inside the container that you're meant to use and scoop your stool sample. So there's a pop-up video that will come up in this video just right here showing you just a brief explanation of how the stool sample will be collected. So this container is going to be labeled with your name, age, sex, and the test, obviously, stool MCS. So the stool sample is meant to be collected on a clean toilet roll when you're in the toilet. And you're meant to use this spoon to scoop the stool, just like the size of a grape. Please, for this sample, you are not to fill up the container. This is a sample that we do not want you to be generous with at all. So once you collected your stool sample, you're now meant to seal the container to avoid any contamination. So once you've sealed the container, you put it in the Ziploc bag we provide for you and seal. Very simple. So once you're done, you give this to the lab technician. The lab technician then takes it to the lab for us to do the dirty work. So once the sample gets to the lab, there are 4 major things we need to do. The first thing we need to do is to check the appearance of the stool. The medical name for the appearance is called microscopy. Microscopy. That gives us an idea, an indication of what we expect to see from the stool sample. If the appearance of your stool is brown and solid, it points towards a normal stool sample. In some cases, it points towards dehydration or constipation. But if the appearance of your stool is watery and loose, it points towards diarrhea or typhoid. The next step is microscopy, which is what we use this for. So the M in MCS is the microscopy that I'm about to explain. So in this step, we look at the stool sample. It goes through some processes which I can't show you now for clear reasons. So we observe the stool sample under the microscope and observe for things that cannot be seen with the eye. Things like parasites, things like bacteria that could contribute to the symptoms you may be having and the discomfort you may have in your belly. Culture is the third step in this process. It's the C in MCS I mentioned earlier. So it involves this thing called the Petri dish. What happens is we take your stool sample and culture it on this plate. We use something called a wire loop. So culture just basically means spreading your stool sample on this plate. After we've done that, we label it so we can be sure this is your sample. Then we put it into this incubator here and incubate it for 24 hours at 37 degrees Celsius. You may wonder why 37 degrees Celsius. There's a reason for this. The reason is that is the temperature that the salmonella grows in your body when you have typhoid fever. If you get it, fever, running a temperature, that's the environment that the salmonella tends to grow. So after 24 hours, we expect to see the salmonella that is causing the problem for you, and we remove it from the incubator. When we remove it, because this is a dummy sample, you won't see anything in this, we expect to see a growth on this plate. there will be a picture that will pop up now to show you what the salmonella will look like. We then perform biochemical tests on that thing to be sure that yes, this is a salmonella, before we now go to the next and final step. The final step is sensitivity, the S in MCS. It involves this new Petri dish. So when we see the salmonella, like I explained earlier, we take the salmonella from this plate and transfer it to this one and spread it across. The magic now happens in this. We now plant different antibiotics on this dish. The antibiotics comes in a disc like this that you can plant like different antibiotics like levofloxacin, ciprotab and whatnot. So after we plant the antibiotics on this, we then place it into this incubator again for another 24 hours. After 24 hours, we then take it out and then observe to see the antibiotics that worked and the ones that didn't work. After all the dirty work, this is the result you typically get from us as your stool MCS result. It shows everything I've explained about. So if we start from the top, you can see the microscopy, which is what we saw with the naked eye. In this case, the patient's result said brown semiform stool, which is just okay. So the step below that is showing microscopy, which is what we couldn't see with the naked eye, under what we saw under the microscope. In this case, it's saying no ova or cyst parasites, saying that means there was no parasites that we saw that could add to the condition of this patient. The penultimate step in this result is the culture, which then shows the bacteria that grows for the patient. In this case, is saying the moderate growth of Shigella and Salmonella. So this person even had two different kind of bacteria that grew in the plate we had. The beautiful part of this test, it now shows the sensitivity, that's the antibiotics that worked, and the resistance of the ones that didn't work. Ones at the very top is 3+, those are the ones that worked the most, while the ones at the bottom are the ones that are not as effective. This is how the doctor will then know what to prescribe for you and what works. The final part of this test shows you how magnificent it is. It doesn't just tell you you have typhoid, it tells you what to do. So why are you still running Widal tests? A wise man once told me knowledge isn't actually power. Knowledge is power with action, when you take action with it. So use this knowledge to pick the right test and not the wrong test. Let's act by ditching Widal tests and doing stool MCS so you can get the right results. If you have any questions, please feel free to call the number that will show on the screen now, or even comment in the comment section. Also feel free to visit us. We don't bite, we're very friendly. We'll attend to you and answer all your questions. Thank you and see you again soon.